Let's start. Hello. Today we have Irbin Veliz. He's going to present his PhD plan. He's in the second year. The PhD is doing here, but also in the University of Sevilla. And it's time to start.
Okay. Thank you, Maria and Javi, for having me. She has said, my name is Irbin, and today I'm going to talk about my PhD plan, titled Evolutionary and Conservation Genomics of Small Mammals in Tropical Southeast Asia, supervised by Dr. Jennifer Leonard from the Conservation and Evolutionary Genetics Group. So first, I'm going to tell you a little bit about me. I did my bachelor's degree in the Complutense University of Madrid, and I had the opportunity to go to Poland to do my bachelor's project, monitoring populations of bats wintering in the Nietoperek Bat Reserve. Afterwards, I did a short-term volunteering internship in the Animal Advocacy and Protection in Alicante, performing behavioral observations of macaques and other duties. Then I did my master's degree also in Madrid in biodiversity and conservation, and for my master's project I analyzed macro ecological data of freshwater ecosystems from the Azores archipelago. And last, I arrived here 3 years ago thanks to a high intro fellowship in the Conceivable group, and then 1.5 years ago I started my PhD thanks to a fellowship from the Ministry of Science, University and Innovation. So in this project, when we see a species, we tend to ask why they are where they are, because not all of these organisms are distributed around the globe the same way. So according to this, we know that there are ecological aspects that constrain a species to a certain area, but we also need to consider the evolutionary history in order to understand how they are currently distributed in the present. So that is why in this PhD we are going to focus on tropical Southeast Asia, because it's an area with a high biodiversity. As you can see in this map, for example, the island of Borneo have the highest species richness. And we are going to combine both field and sequence data to study ecological and evolutionary hypotheses using non-volant small mammals as a study systems. But in particular, we are going to focus on the mountain tree shrew, which is this little animal here, known as Tupaya Montana. This is a Bornean endemic, and it is a mountain specialist. The reason why we are focusing on this species is because it has a broad elevational range, something that is not common among organisms within the tropics. It is omnivorous, and a mutualistic relationship with the pitcher plant has been described. As a curiosity, the local refers to this animal with the word tupai, because in Malaysian it means a squirrel, and as you can see, because of its external appearance, it resembles a squirrel, and they have scansorian traits, which means that they are prone to climbing, but they don't need to. The last time that it was assessed, it was in 2016, and they were classified as of least concern. More things about these animals. Well, this animal. If you see these pictures, it's no wonder why scientists from the past didn't know where to put them, because they look alike like primates or rodents. But actually, they belong to their own order, which is the order Scandentia, and it's closely related to primates and not to rodents, as it could have looked just by the external appearance. This is also important for the following slides that I'm going to talk about. Now, let's move to the study area. These animals were sampled in the Kinabalu National Park in Sabah in Borneo, and this park has 2 mountains, Mount Kinabalu and Mount Tambuyukan, with different peaks. Also, for Mount Kinabalu, 4 vegetation zones were described, and the Tupaya Montana can be found in these 3 vegetation zones. Regarding the ecological aspects of this mountain, for Mount Kinabalu it was described a reduction of temperature of 0.55 degrees every 100 meters of elevation gain. So, in the first chapter, as I have mentioned before, I'm going to focus about what drives the range limits of a species. If you can see that here, the species range limits is essentially the expression of a species' ecological niche in a space. But little is known about why, like the differences that can be on populations on the edges and the ones on the center of distribution. For example, it might be that the populations from the edges exhibit less diverse immigrants, but also they receive a lower amount of immigrants, as you can see in this figure. It could also happen that on the edges, the selection is different than on the center populations. For example, here we could have directional selection, and here we could have stabilizing selection. Last, also populations from the edges could experience greater rates of survivability and reproduction due to experimenting more strong limiting factors. There are three different theoretical categories that have been trying to explain the origin of range limits. For example, there are those that do not incorporate any environmental heterogeneity information, and they explain the range limits of a species through a Lie effects, which means that a population cannot grow if it's too small, or ecological interactions such as interspecific competition. If we incorporate the environmental heterogeneity to these models, then range limits will come determined by the fundamental niche limitation, or also dispersal disequilibrium due to barriers. Last, we have models that incorporate both the environmental heterogeneity and also the evolution factors. And then here, if genetic variability is enough, the species will be able to adapt to the next habitat. So our question is, what drives the species limits? And we are going to focus on the abundance centre hypothesis, which states that in the range centre we can find the optimum conditions for a species to live. Therefore, there will be a high abundance of individuals, which could be translated as a high effective population size. And this bigger population size could mean that there is higher genetic diversity than on the range margins, since where the ecological conditions are not that optimum, the abundance will be decreasing until a point where the species does not exist anymore. And this will also mean that genetic diversity decreases from the center towards the edges. But there is also another explanation of why a species is not able to move to the next habitat, and that is the gene flow from the central populations to the populations from the periphery. And this gene flow will be swamping out any possible adaptive allele. To test this hypothesis, as I have mentioned before, we are going to use the Tupaya Montana, since it has a broad elevational range and it inhabits different habitats. We consider that the range center is located at mid elevations, and there is where we expect to find a higher relative abundance of the individuals, and also a higher genetic diversity. And as we go down or to the top of the mountain, we will find less abundance of individuals and less genetic diversity. In order to do this study, we gathered data from two previous studies. We took the traping data from this study, and with this traping data, we calculated the relative abundance per transect elevation. There were 6 transects per mountain, and the relative abundance expressed as a percentage was calculated as the number of mountain trees you trap per transect elevation divided by tetrapnites per transect. With the sequencing data, we are going to use we have used ultra-conserved elements, which are highly conserved regions across the genome. And they are important because, as you can see here, they are highly conserved across different taxa. You can see here, for example, how these organisms share the UCs. They are also present in high numbers throughout the genome. And they are informative mainly because of the variability that can be found on the flanking regions. In the core region of the UC, we can find some variability, but the density is not that high, as you can see in this graph. And the function of the UCs is still under study, but they might be regulatory or enhancers of gene expression. In order to obtain UCs from a raw sequence data, there is a pipeline developed by the Fairclough lab, and it's called Filuche, because they have been used in phylogenetic studies, but also in population studies. So some results that we have obtained, as predicted, the highest relative abundance was found on both mountains in these habitats, in the lower montane and upper montane forest. For the Mount Kinabalu, this corresponds to the center of distribution, but for Mountain Buyukon, this doesn't, but because that's where the mountain reaches the top, I mean, you cannot go higher than that. Then, for the genetic diversity, sorry, I forgot to mention that we use the ultra-conserved data in order to estimate autosomal heterozygosity. So with autosomal heterozygosity, we found that from the lower elevations to the higher elevations, genetic diversity decreased significantly in Mount Kinabalu, but there was no apparent trend in Mountain Buyukon. So if we go back to what we have said before about how the hypothesis was that at the edges of a distribution, the low genetic diversity will be the determinant of a species not being able to adapt to the next habitat, we can see here that that is not the case, because genetic diversity was not lowest at the periphery, but there was a decrease, actually, if you went up high to the mountain. So that's not match the hypothesis. So if genetic diversity is not what is driving in this case, the lower distribution limit, then it could be gene flow that is swamping out any adaptation on the edges. But so the question comes from also the fact that there is still habitat available in the lower limits, but this species does not go down below 900 meters. And we want to know why. Another explanation could be the low individual fitness on the edges of the distribution or range. And that takes us to the second chapter, in which we are going to try to estimate fitness using genetic load as a proxy of this parameter. Genetic load is defined as half-form genetic variants produced by deleterious mutations. And in diploid species, it can be divided into the realized load, which is the one that is actually expressed, and it affects reducing the fitness of present generations, and also mask load, which is the possible reduction in fitness due to if these deleterious alleles become expressed in an individual. And also, I need to mention that purging can act only on realized load because it's the one that is expressed. So for this chapter, the hypothesis will be that on the edges of the range of this species, so that will be near the 900 meters distribution or up to the mountain, the genetic load will be higher, but on the center, the genetic load will be lower. How are we going to do this? Well, we are going to use the same raw sequencing data from the previous chapter, but in this case, we are going to use the human reference genome and the human cat scores in order to get an estimation of the genetic load. Cat CAT score, well, a CAT is a framework where that means that it's combined annotation dependent depletion, but it is a model that ranks single nucleotide variants and assigns them a deleteriousness. And in order to assign this deleteriousness, it is based on different genome annotations, but also it's nice if you can incorporate evolutionary conservation. And that is why the UCs are useful in this case, because as I have mentioned before, they are highly conserved across different taxa. As you can see here, it has only been developed for human, mouse, and pig, which means that this is something relatively new. So, this is the pipeline that I'm partially following, which is from this study that it has not been published yet, but there is a preprint available if you want to read it. But in this case, we are kind of omitting this part because we do not have a reference genome for our study species. What we are doing is, from the human reference genome, we have extracted the ultra-conserved regions, and with our raw sequencing data, we have mapped our data to the human reference genome in order to call variants. And once variants have been called, then we can lift over the CAT scores of the human genome and pass it on to the UCs of the Tupaya Montana. I'm in this state, but it's still something that I need to figure out. So once you have managed to rank all the variants, you will need to filter and remove the sites that are non-scored or that are fixed. And then genetic load will be the sum of all homozygous loci and half of the heterozygous loci for an individual. And this is because for the heterozygotes, this 0.5 factor refers to the probability that a deleterious allele is passed on to an offspring. So a summary of the first and second chapter is that we have been investigating what is driving the species range limits. And for that, we have both look at the genetic diversity and the genetic load. We know that our study species occupy different habitats, so we suspect that there must be some kind of local adaptation. And that takes us to the third chapter. The third chapter is still being developed, and it might be split into two chapters. But again, a reminder, the Tupaya Montana goes from 900 meters up to 3,400 meters. And there are these different habitats. So previously in this study, it has been described phenotypic variation in these animals, and they found that individuals from the range center were the smallest among the whole range distribution, and also that individuals from the upper mountain forest had a higher length on the rump and scapula. So another example, this time in a temperate species, is the deer mice, Peromyscus maniculato, that also has a broad elevational range. And they have found in this animal that, for example, this gene is a candidate gene for hypoxia adaptation. What we want to do here is address this matter through two different approaches. The first one will be to look for genetic basis of local adaptation using candidate genes. For this, we will generate a new dataset resequencing individuals from the whole elevational range. And yes, we are going to try to search for signs of differential selection. Another objective of this chapter will be to assemble and annotate a reference genome for the mountain trees group. This could be a possible collaboration with the Uppsala University, and with the data generated, we will perform genome-wide association analysis. Here you have how the process of assembling a genome is done. And so, yes, that will be all. Thank you to the Conceivable Group and also to the Spanish Ministry of Science, Innovation and Universities for this opportunity. Also, I have a video of the organism because I believe not many of you have seen it, so I want to reproduce it. I will. Okay.
Forests are almost always cold and wet. Here in the mountains of Borneo, the shrew has had to make peace with a killer to eke out his living. Although a carnivorous plant by nature, mature Lowe's pictures attract guests for more benign reasons. It seduces its visitor with sweet nectar. The shrew has something it needs. The pitcher has modified its design to position its fleshy bowl right under the feeding shrew's rear end. While its diner is busy, nature takes its course. The shrew gets its sugar hit, and the Lowe's pitcher collects its reward. Nitrogen-rich shrew poo. Teaming up has allowed both pitcher and shrew to solve the problem of finding enough food in the cloud forest. While the shrew is preoccupied, he's being watched. The activity has caught the attention of a leopard cat. With so little prey around, this is a tantalizing opportunity. Agile reflexes, and the game of cat and mouse.
Thank you very much, Irbin.
Wait.
It was really interesting.
Okay.
Thank you. So now it's time of questions. One.
Okay. I like the video. No, the video was very nice, thank you. But precisely that brings to one question, that the patterns that you're studying of genetic diversity, you are assuming that the physiology is the limiting factor. But why not the plant? Why not other factors could be the limiting, that maybe there is a physiological limit, but at the same time, in the center of the distribution, maybe it's not suitable for large numbers of this plant or other resources.
Yes.
Maybe.
The population density and the ecological limits are not so well correlated. So do you have any information about this, or what is the preliminary data suggest?
Yes, I believe that in order to study a species, you cannot focus only on this species, but also have into account the biological interactions with other species. And for example, in the lower, like below 900 meters, there is no Tupaya Montana, but there are other species of Tupaya. So that might be, there could be some interspecific competition. Also, regarding the upper ages, it's because, at least in Tambuyucon, there is no more mountain, but I am not sure about, maybe it's also like in Kinabalu, since it reaches the 4,000 meters, and the range of this species ends up around 3,400, that the habitat there is not suitable, so we could also look into that, but we are mainly...
Yeah, but it's going to be very interesting to try to correlate that with also with the population density with the data that you're gathering. That's very interesting. Mm-hmm. Thank you.
Yes, I believe.
Yeah, so I have a couple of questions. One more general, in the sense that maybe related to that. I mean, it seems like addressing the limit problem, what's limiting the range, and it has to do more on ecological, on ecology and biological interactions and with the media rather than genomics, right?
Yeah.
So is there anything done, or is there anything you're planning to do yourself to address this? What's ecologically limiting the range of these species?
Well, for example, with the temperature, this species experiences a large, how can you say it, temperature range, because if temperature decreases from like 0.55 degrees every 100 meters of elevation, in another study they reported that the thermic range that they experience in Mount Kinabalu is from 12 degrees, and in Mount Tambujucan of 8 degrees. So, but in this other study where I mentioned the phenotypic variation, they reported that temperature might not be the main driver of selection, because it seems like these species have adapted phenotypically to the environment, so this temperature range wouldn't be of such importance. And also it has to do with the hair length and how they can build burrows in order to mitigate these temperature changes. Regarding other aspects, I guess we could look into the ecological niche or trophic niche. We actually were considering using stable isotopes in order to look at the whole non-volant mammal community in these areas, but depends on how this goes, and if there is enough time, then that could also be another possibility.
No, because if the limit is more exogenous, I mean, ecological
Yes.
or interactions, biological interactions or so, then any pattern you see in genomics could be rather the consequence of that, rather than the cause of the limit, right? And my other question is kind of more technical. Why the focus on ultra-conserved elements? Because you're, at least the latest
yes.
chapter, you will probably need more extensive genomic information, so why not starting with that?
The focus, first I will say it comes from, because on this study, from what I gathered the raw sequencing data, these DNA sequences were enriched for ultra-conserved elements, because they wanted to test if they could use them for population studies, but also the ultra-conserved elements have an utility because as I have said, they anchor genetic markers on the flanking regions that can be used for both phylogenomic and genetic studies. But also, since they are found in numerous numbers across the genome in very different taxa, that is also very informative.
Okay, but in terms of getting the information, is there any, I mean, because you're saying because the information is already available, the data was already available, or it's just more easy or cheap to obtain?
I don't know if it's easier to, like if it's cheaper to obtain, but yeah, it was mostly because the data was already there, so we wanted to analyze it in a different way.
Thank you. Good luck. Yeah.
Thank you for your talk, Irbin. Well, I have two suggestions, but well, it's related to you are working in a mountain, and the mountain is a conic shape, has a conic shape.
Yes.
So you have that lowland areas has more extension than the top.
Yes.
So I don't know if this may have something to explain, something related to maybe cause or indirect or direct or anyway, to explain the genetic pattern or decreasing with altitude in the Kinabalu, which has a larger span of altitude. And I don't know if this has been shown in the literature, if altitude in tropical mountains or in other mountains, if this decrease the genetic diversity, have the heterozygosity.
Yes, I believe that, I mean, there are cases in which genetic diversity decreases with elevation, but it's not a general pattern. So there are cases that genetic diversity can increase through elevation, but it has also to do with the evolutionary history of an species. For example, it could be that this species is expanding, so then the signatures will be different. But for the Tupaya Montana, in this other study that I have mentioned, they first established in Mount Tambucon because it's the older mountain, and then it seems like they moved to the Mount Kinabalu. So that might be also why genetic diversity is lower at the top of the mountain.
Yeah, also the mountains have, as you know, that the higher diversity at the lowland and the top. And the top
Yes.
there is a vacuum of species. So, well, that is very speculative, as you can imagine. My second question is related to the physiology, because you are talking about physiological limit and so. That's what you have to show, or you have to because it's a very adequate hypothesis, but you have to test anyway, because when you test, probably you discover that physiology does not explain anything. Yes. But, well, one possibility, if you try to make some inference on this physiological stuff, is to measure, for instance, what is called the thermal neutral zone, that is typical in homeotherms, where the metabolism is stable in a range of temperatures. When you are at lower temperatures of this range of thermal neutral zone, or colder or higher, the metabolism increase speedily. So if you have the possibility to measure metabolism and to discover which is the range, altitudinal range, or sorry, temperature range, because you are in a mountain and a tropical mountain where the variation is only altitude, elevation, nothing more, and habitat, but probably are more woodlands and so you have no open area, so probably the variation in temperature is only elevation. So if you have, you can correlate to temperature with elevation. So you have the range of temperature in the range of elevation, and then test if the animals fit to this range of temperature or not. And then you can test this possibility of physiological constraint to determine the range of distribution.
Yes, I think that's a nice thing to do. I was going to comment that, also in the study of phenotypic variation, how they reported that body size was lower at the intermediate levels. They also mentioned that in general body condition, which is like a proxy of the welfare state, I'm not very sure about this, or something to do with the lipid content, did not, if I remember properly, did not change. So that is showing the local physiological adaptation, I will say. But as you said, we need to explore that. I think I don't know if we will. Yes.
Thank you. Thank you.
So I wanted just as a curiosity, how does this animal move? Does it move a lot throughout the gradient? They are kind of
Yes. From what I have ranged, the home range is of 160 meters, but also it has been suggested that along the mountain, if I remember correctly, on the lower elevations, they are not as scansorial as in the upper elevations, and this might have to do with the food availability or resource availability, because as I have said, they are omnivorous, which means that they can feed on fruits, but also insects, so yes.
And are you planning to measure more traits, or are you gonna or are those traits already gathered?
For this species, they for this data set, they have already analyzed for example, the length of the tail, the body size. So if we want to do more analysis regarding this, it should be, I believe, in other parts.
Okay, so that's all my questions. Any more?
Thanks, Irbin. Regarding chapters 1 and 2, do you think you have because I think you might have the risk of, even if the questions you are setting are pertinent from an evolutionary point of view, I think the processes you want to measure maybe are so subtle that the homogenizing gene flow in the population might obscure those processes. Do you think you have enough resolution in terms of number of individuals or SNPs to measure it, or is there any power analysis you could do, like using maybe some spatially explicit models to test it? Yeah.
To test the...
Because on the one side you have gene flow that is homogenizing the genetic variants across all the elevations, and on the other side you have at the edges where there are changes in allele frequencies going on because of given factors that you want to test. So I don't know if you have enough power to test them. I don't know if you think.
Yes, with the ultra-conserved elements, I am not sure if they are powerful enough to look at these processes, as you have said, since they might be obscuring the little differences. So I might have to think about it. Yes.
Any more? Okay, so that's all. Today was a short seminar. See you next week with 2 more PhD students. Thanks. Thanks, Irbin.